In this video tutorial, I am going to discuss about the secondary structure of proteins. Now, in the primary structure of protein, we know that primary structure of the protein is the simple sequence of amino acids. Now, in case of secondary structure, this amino acid sequence is regularly arranged. This peptide backbone, it does not assume any random 3D structure. There is some kind of regular arrangement. So, there is a regular arrangement of amino acids occur. Regular arrangement of amino acids. Now, which amino acids? Amino acids which are located near to each other, which are neighbor to each other. Suppose this amino acid, its relationship with its first neighbor, second neighbor, third neighbor, fourth neighbor, but not these amino acids relationship with the 100th neighbor, right? So, it is a regular arrangement of amino acids that are located near to each other, right? Located near to each other in the linear sequence. So, this is the definition of the secondary structure of protein. What is secondary structure of protein? It is the regular arrangement. It is not any random arrangement. It is the regular arrangement of amino acids that are located near to each other in the linear sequence. So, there are many different types of such regular arrangements are seen, okay, as in secondary structure. What are these different types of regular arrangement? So, the first and most common is the alpha helix. Then the second type is the beta sheet, which is also called as beta pleated sheet. Beta pleated sheet. Then the third type is the beta bands. Beta bands, which is also known as beta turns, or other name is reverse turns. Reverse turns. The fourth type is non-repetitive structures. Non-repetitive structures or arrangement, okay, or secondary structure. Non-repetitive secondary structure. And the fifth type is the super secondary structure. Super secondary structure. This is also called as motifs. Now, all of these different secondary structures, they are all stabilized by the hydrogen bonds. All stabilized by hydrogen bonds. This one thing is common in all of this secondary structure, right? The second thing which is common to all is that whenever we want to study any of this structure, we have to use one specialized technique, that is circular dichroism spectroscopy. So, technique to study is the circular dichroism spectroscopy. So, by using this technique, we can study any of this secondary structure. So, in this video, I will be discussing about the alpha helix. The discussion about this remaining structure, that I will take up in the next video. Okay. So, let us start our discussion for the alpha helix. So, alpha helix. So, in case of alpha helix, there are many different types of polypeptide helices are seen, but this alpha helix is the most common. Okay. So, we can say that several different polypeptide helices are seen. Helices are there in nature, but our alpha helix, it is the most common variety. Okay. So, that is why we will study alpha helix in a detail. So, now the alpha helix, it looks like this. It is a helix, and this helix is the right-handed helix. So, right-handed helix. This helix, it is not a flexible one, but it is a rigid one. So, here what I had done is, I had shown you the ribbon diagram of the alpha helix. Now, in this ribbon diagram, this ribbon is made up of polypeptide backbone, but whatever the side chains are there, these side chains, they project outside of this alpha helix like this, okay, like this. So, what happens? The carbon, this is the peptide bond, right? This is the peptide bond. So, that peptide bond and its backbone, it is in this helix, but whatever the side chain of this alpha carbon is there, they project outside of this helix. It does not project inside of this helix. Now, why? Because if all the side chain they project into the inside, what will happen? There will be a sterical forces, and this sterical forces, it prevents its thermodynamical stability. So, to maintain the thermodynamic stability, what happens? This side chain R, they project outside of this helix. So, we can write that side chain of amino acids, are extended outwards. Why? Because there is only a limited space available inside. So, why they are extended outward? Because to avoid interfering sterically with each other. Now, how this helical structure is possible? So, this helical structure, it is possible because of the extensive hydrogen bonding. Now, how this hydrogen bonding formation takes place? For that, I will take up a new diagram. So, this is the same structure as the ribbon structure, okay? But here what I had done is I had shown you all the atoms inside this. So, as you can see, these all are hydrogen bonds. This dotted line, which is I highlighted in the yellow color, these are all hydrogen bonds. So, there is extensive hydrogen bonding, it is seen. Now, this hydrogen bonding is between which molecule? As you can see, at every place hydrogen bonds it between this oxygen and this hydrogen. It is here also oxygen and hydrogen, oxygen and hydrogen. So, this hydrogen bonds, it is always between oxygen and hydrogen as far as alpha helix is concerned, right? Now, is this any oxygen? No, this oxygen, it is attached with this carbon. So, we call this oxygen as a carbonyl oxygen. So, carbonyl oxygen, and this carbonyl oxygen, it is a part of peptide bond. So, we can call it as a peptidyl carbonyl oxygen. Again, is this hydrogen any hydrogen? No, this hydrogen is the specific one. This hydrogen is from this amide, okay? So, this hydrogen, it is the amide hydrogen. Now, is this any amide hydrogen? No, it is a part of this peptide bond, right? So, we call it as a peptidyl amide hydrogen. So, this hydrogen bond is between two specific molecules. One is the peptidyl carbonyl oxygen and second one is the peptidyl amide hydrogen. Now, these both are different. This oxygen cannot make hydrogen bond with this hydrogen, but it has to look for another amide hydrogen which is part of another polypeptide, another peptide bond, right? So, we can say that this hydrogen bond is between the one peptidyl carbon oxygen with another peptide bonds peptidyl amide hydrogen, right? This hydrogen bond, it is parallel to this helix. Suppose this is the central axis of this alpha helix, then all these hydrogen bonds, they are parallel to this central axis of alpha helix. So, we can say that all the hydrogen bonds are parallel to the spiral. Now, see, here the hydrogen bond is between this and this. So, if we count, then this is the first peptide bond, then this is the second peptide bond, this is the third peptide bond, and this is the fourth peptide bond, right? So, this hydrogen bond, there is a spacing of 4 peptide bonds. So, spacing hydrogen bonds, there is a spacing of 4 peptides, right? Or you can also call it as a 4 amino acids. Now, see, this is the starting of peptide bond, right? This is the N terminal, whereas the lower part, this is the C terminal. As you can see, so here all the amino acids are participating in the hydrogen bond formation except for this first amino acid and this last amino acid. See, in this first amino acid, on this side there is a peptide bond, but here as this is a N terminal, there is a NH3 plus. This amide hydrogen is not participating in any hydrogen bond. Similarly, on the C terminal, the last group is going to be a COO minus. Okay, carboxyl group is there. Now, that oxygen will not be able to make up a hydrogen bond. So, all the amino acids, they make up a hydrogen bond except for this first and last amino acid. Now, the last but not a least, that each hydrogen bonds are weak. Hydrogen bonds are not stronger one. Hydrogen bonds are weak, but collectively they are so strong that they maintain this rigid alpha helical structure. Now, if we talk about the dimensions of this polypeptide chain of this alpha helix, then we can find that each turn, suppose if you consider 1 turn, 360-degree rotation, this is the one rotation, there is approximately 3.6 amino acid per turn. If we consider this much turn, which is the 360-degree rotation, in each turn there is a 3.6 amino acids, right? And if you measure a distance, this much distance which is travelled by alpha helix by making 1 turn, this much distance is the 5.4 angstrom per turn. You can also call it as a pitch distance. Okay. Now, which kind of amino acids will be preferably found in the alpha helix, that we need to discuss. So, for that I will take up third diagram. So, in this, suppose this is the N terminal. Okay, that means there is a free amino group is there, and this is the C terminal. So, now in this diagram, see, there is a proline and there is a glycine, which never found in any alpha helix. Why proline is not found in the alpha helix and why glycine is not found in the alpha helix? The reason is that in case of proline, suppose if we see this diagram once again, the structure of proline is like this. So, there is unusual kind of structure is there in case of proline, right. So, this is the structure of proline. Now, in case of proline, what is the problem is this alpha carbon and this amide nitrogen with its hydrogen, they both are part of this 5 membrane ring. So, what happens, this bond, it becomes rigid. Now, once it becomes rigid, then what will happen? In case of polypeptide chain, there will be a sharp turn will be seen. Like suppose this alpha carbon is over here, right? Then what will happen? There is a nitrogen over here. So, this N, now it needs a smooth curve, but instead of that, what will happen in this case? There will be a direct sharp curve like this, okay, and then there will be a hydrogen, and when it makes up a peptide bond, so what will happen here? It makes up a peptide bond like this. So, this hydrogen will be lost while making peptide bond. So, here two problems are there. First problem, there will be a sharp turn over here. So, it will disrupt this alpha helix, that is the first problem. Second problem, there is no any hydrogen attached with this nitrogen. So, it is a lacking of amide group. So, what will happen? This nitrogen, now it will not able to make up a hydrogen bond. So, we know that in case of alpha helix, any amino acid, they have to make hydrogen bond, then only there is a place for that amino acid in the alpha helix. So, because of these two issues, first is the sharp turn, and the second issue is no hydrogen bonds, right. So, because of these two issues, proline cannot occur in case of alpha helix. Now, we understood that why proline cannot occur in the alpha helix, but why glycine does not occur in the alpha helix? The problem was it was too much rigid, right, too much rigid, whereas the problem with glycine is that it is too much flexible. So, in case of alpha helix, we need a smooth turns. So, whenever glycine will occur, that portion will become too much flexible. So, there will be a bending of alpha helix will occur, which will again disrupt the alpha helical structure. So, that is the first issue. Second issue is that long block of positive charge amino acid, long block of positive charged amino acids. Now, this is not possible in case of alpha helix. Why? Because suppose you have long block of positive amino acids, positive charge amino acid in this alpha helix, what will happen? Their side chain will repel each other. So, alpha helix will not be stable. Again, the same thing is true that long block of negative charge amino acid are also not possible in case of alpha helix because of the same reason. If long block, suppose positive, 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 all they have plus positive charge side chain, then they will repel each other. The third is long block of bulky amino acid side chain, bulky amino acid side chain. Now, this is also not possible because again, if there is a long block of bulky amino acid side chain, then there will be a steric collision with each other. So, again this is also not possible in case of alpha helix. One thing is favorable, that if suppose we have one positive side chain containing amino acid over here and one negative side chain containing amino acid over here, and we know that this is a complete turn. So, 3 to 4 amino acid apart, if there is one positive charge amino acid, then it is more likely that one negative charge amino acid will be there after 3 to 4 amino acid. Why? Because they will occur near to each other when this helix is formed. So, this is favorable condition, right. So, positive charge and negative charge R groups tend to occur 3 to 4 amino acids apart, right. As we had discussed earlier that each turn it contains 3.6 amino acids. So, that much spacing is required. The same thing is true, the same spacing is required in case of hydrophobic amino acid, hydrophobic side chain amino acid, right, side chain amino acid. Suppose this is hydrophobic amino acid, then if this is also hydrophobic amino acid, then there will be a matching group. So, what will happen? They can carry out hydrophobic interaction with each other. So, hydrophobic amino acids tend to occur 3 to 4 amino acids apart. Now, last but not a least, that is on the N terminal, we know that there is NH3 plus group, right, and on the C terminal we have COO minus group. So, what will happen? There is a dipole like moment. There is a positive charge over here and negative charge at this end. So, what will happen? To neutralize this positive dipole at the N terminal, more negative charge amino acids, they tend to stay on this N terminal, and to neutralize this negative charge at this carboxy terminal, more positive charge amino acid tends to accumulate over here. So, these are all characteristic of the alpha helix. Now, what are the examples of this alpha helix? Which protein they contain more alpha helical structure? So, we have two example. One is keratin and second one is the myoglobin. Now, keratin, it is the rigid fibrous protein. It is a rigid fibrous protein, whereas myoglobin, it is a soft or flexible, it is a flexible and globular protein, right. So, here you can see that both the proteins, their physical properties are vastly different. On one side keratin is there, which is rigid and fibrous protein. On another side myoglobin is there, which is flexible and globular protein. And in both of these, keratin as well as myoglobin, extensive alpha helical structure is seen. Alpha helical structure is seen. And you can say that this keratin is almost 100% of the structure of this keratin, it is made up of alpha helix. So, this is all about the alpha helix. In the next video, we will discuss about beta sheets and beta bands. If you have any query or confusion, please write it down in the comment section below. Thank you.